Okay, let's look at glomerular filtration, one of the most important parts of what's going on in the kidneys. And of course, the functional unit of the kidney is the nephron. And remember, there are probably about 1 million nephrons in each kidney. So we are actually in the, functional unit area. Part of that functional unit is this renal corpuscle, which was made up of the glomerulus, this capillary here, and the Bowman's capsule. And then, of course, we can see the efferent arteriole here, where, we're sending blood in and blood out of here, and we are filtrating the contents of the blood into this capsule to form this product called filtrate, and then we're going to send it to the proximal convoluted tubule. So hopefully that makes sense so far. But what we're really wanting to understand on this is what's going on in here, and also, this is really important, the three main processes of what the kidney is doing is filtration, which is going to basically be happening here in all these renal corpuscles at the glomerulus. Reabsorption, because most of what we filter out has to be reabsorbed back into the blood, or we would be dead in just a couple of hours if we didn't reabsorb most of it. And secretion, which is a very minute but important function that's going on before we finish turning the filtrate into urine. Okay, so let's continue and talk about this. A couple of things that, it helps to know going into this is, first of all, what we already mentioned, the nephron being that series of cells that make up the Bowman's capsule, make up the glomerulus, so that's the renal corpuscle. Of course, we've got the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and the collecting ducts. And then we have the peritubular capillaries that are, kind of surrounding those tubes so we can reabsorb the filtration. So if you're not familiar with that, go back over the lab material and look back over that. Okay, now the next thing that we need to talk about, though, is the difference in hydrostatic pressure and osmotic pressure. Hydro means water, and of course, osmosis has to deal with water movement, but so hydrostatic pressure and osmotic pressure both have to deal with the pressure of water, but it's two different types of pressure, and the best way I can describe it is hydrostatic pressure would be kind of like the pressure of water in a water hose. How fast is the fluid moving through here, and how forcefully is it leaving here? Okay, whereas osmotic pressure was the pressure of how water moves in a either isotonic, hypotonic, or hypertonic solution. If you remember, water always goes from a high concentration to a low concentration, and that's our definition of osmosis. It is the diffusion of water. So whatever our concentration is of the water, in other words, do we have a hypertonic solution, especially when we're comparing the solution of the filtrate to the solution of the blood, then those are going to be pressures that are going to be involved in understanding our glomerulus and understanding the higher pressure that we have to exert in this area, and for those reasons, which we'll get into. So again, even looking at this picture, we again see our renal corpuscle, the glomerulus with the Bowman's capsule. We see the proximal convoluted tubule. We see the afferent arteriole, this sending blood in here. We see the efferent arteriole. Notice how the efferent arteriole, the diameter of it's bigger. The efferent arteriole, the diameter is smaller. And then, of course, what we're doing, we're filtering the part of the plasma and part of the contents of the plasma, and it's becoming something called filtrate, and then it's running, and that's our first step, filtration. It's running through the proximal convoluted tubule with these cells that have what we call brush borders on them because they've got microvilli on them. And then what we're going to do is we're going to reabsorb the majority of what we filtered out back into the peritubular capillaries, which is what the efferent arteriole becomes. So remember, what we filter out, we're going to reabsorb most of it back into the bloodstream. Keep in mind, though, even when we're filtering it back in the bloodstream, there's still interstitium between the cells of the tubules and the cells of the capillaries here. So here we actually can see, and let me make this a little bit more clear, we can see the different parts of the nephron. Of course, this is a blown up version of what we have here. And here we see our proximal convoluted tubule, our loop of Henle, our descending loop, our ascending loop. Here we see that the ascending loop then becomes the distal convoluted tubule when it actually, so here's the ascending loop at this point here where we're going to form a structure called the juxtaglomerular apparatus. At that point then it will extend on past that and become the distal convoluted tubule and run over here to the collecting duct. Again, notice how the efferent arteriole is wrapping back around and paralleling the it's becoming the peritubular capillaries and kind of paralleling the tube for this all-important reabsorption, and also for something called the countercurrent multiplier and exchange system, which we'll mention later. All right, so let's keep it rather basic right now and talk about what's going on. So again, here are the parts we talked about: the afferent arteriole, the glomerulus being the capillary, the capsular space being the space between the capillary and the capsule, and of course the glomerulus and the glomerular capsule together make up the renal corpuscle. Again, we see the efferent arteriole. Notice the diameter again of this is bigger, the diameter of this is smaller. Notice we actually have our proximal convoluted tubule coming out, and that continues on as the rest of the tubules. Here again we have our peritubular capillary where the efferent arteriole is pretty much paralleling the tubule so it can reabsorb the majority of what we filtered out. Okay, here's that first step again where we're literally are filtering whatever's flowing through here is leaking out and becoming this material called filtration, or excuse me, called filtrate, and that process is called filtration. Now we'll talk about what is able to get out here and how does it leave the capillary and become the filtrate. We'll be talking about that in the near future, but right now we're just saying the process of it leaving at the inside the capsule and becoming filtrate is filtration. Again, remember this is happening in a million of these in each kidney. Okay, so filtration was happening here, but notice when the filtrate goes into the tubules, now we need to reclaim the majority of what we filtered out because it was mostly size and charge dependent as why it filtered out. It didn't mean that everything that filtered out was stuff we wanted to get rid of. Now the waste products we're getting rid of, most of it did happen to be the correct size and the correct charge, but we also filtered out a majority, a big amount of things that we can't afford to lose or will die. So notice the arrows are showing that what was filtered out is now being reabsorbed back into the bloodstream, going from the what's called the lumen of the tubule through the cells in cellular transport into the interstitium, and then from the interstitium back into the bloodstream. So this again is this important process called reabsorption. Okay, notice our third step is called secretion, and what we're doing now is we're having very minute adjustments of product that was still in the bloodstream that didn't get filtered out initially, that still we don't want to keep in the blood, but that we want to get rid of. So we have basically one more chance to get rid of products, even though, again, this is a very, very small amount compared to what was filtered out and compared to what was reabsorbed. But it is our last chance to make some adjustments to what we want to get rid of. Now, we haven't really talked about the adjustments in the water content, but as far as what's in the filtrate that's becoming urine, here's our last chance to basically fine-tune that. Okay, the pressure of the blood coming in and the fact that this afferent arteriole is normally a larger diameter and this efferent arteriole is normally a smaller diameter, the pressure of that coming in is going to push blood through the capillary. Well, in the meantime, we literally have three things that we need to be able to cross. It's called the filtration membrane that we need to be able to cross to get out of the lumen of the capillary and into the Bowman's capsule space here. So what we have to do is we have to basically, if you'll notice, here is the inside of the capillary. So here's plasma and here's a red blood cell, but the contents of the blood that we're filtering out have to go through three parts of the membrane. Okay, the filtration membrane, the three layers it has to go through is, first of all, fenestrated glomerular endothelium. Now remember, the actual simple squamous epithelial cells that line a capillary are called the endothelium, and fenestrated means that it has little bitty holes in it. Little bitty holes that if you're bigger than those little bitty holes, you should not be able to leave this. So a red blood cell or proteins should never be able to leave the blood, because first thing, they shouldn't be able to go through that fenestrated glomerular endothelium, the little holes in the squamous cells that line the lumen. Okay, now the next thing is the basement membrane. Remember, epithelial cells are always connected to other tissues or to other epithelial cells by a basement membrane. So we have to have a porous basement membrane. So you got to go through the little epithelial cells. You got to be able to go through that basement membrane. And finally, there's something called filtration slits, which are formed, as this says, by the pedicels of the podocytes. Now, the podocytes are the cells that make up the outside of the capillary. So these are very specialized capillaries. They're not just simple squamous epithelial cells like most capillaries. These are specialized capillaries. They've got the endothelium, which is more like a normal capillary, except it does have the fenestrations or the holes in them. Then it is connected to this outer layer of cells called a podocyte. And this outer layer of cells has what they call fringe-like extensions. And the fringe-like extensions are almost like little fingers that are outside. And where they overlap with other fringe-like extensions, we literally have little space between it. So we call those filtration slits. So you've got to go through the holes. You've got to go through the basement membrane. And then you got to go through where the overlapping pedicels of the cells called podocytes we actually use the term interdigitate, like putting your fingers together from one hand into another and then seeing the space between the fingers. Well, the space between the fingers would be the filtration slits. So again, you've got to be pretty small to be able to cross all three of those things. And so we know that we should never find big items in your urine, or something has damaged this area to allow large things out.
Of glomerular filtration.
Okay, notice now.
Glomerular filtration is a process of bulk flow driven by the hydrostatic pressure of the blood. As you can see, small molecules pass rapidly through the filtration membrane, while large proteins and blood cells are kept out of the capsular space.
Okay, so we see these large things being kept in, but we see the small things passing through those three, the three things we just talked about, the glomerular endothelium with the fenestrations, the little holes within the endothelium, the basement membrane, and then finally the filtration slits, which are basically found where pedicels interdigitate from the podocytes, the cells. Now, I know that's a lot, but the main thing you need to know is it goes through fenestrations, the basement membrane, through the fenestrations of the endothelium, through the porous basement membrane, and through the filtration slits of the outer cells, which would be the podocytes. Okay, what are we actually filtrating? What is it that is able to go through that filtration membrane and become our filtrate here in the space, the glomerular space? Okay, so obviously water is part of it, because blood is mostly water, and this is plasma, was mostly water, so the water's carrying all the other stuff out. Where ions are small enough to go through this, so sodium, potassium, and chloride, which we can afford to lose some of this, but
filtrate is similar to its
we can't afford to lose all of it. Now, here's what we're really looking to get rid of in the water is the nitrogenous wastes. These are the wastes that the cells produce that the blood picked up. But if we keep these wastes in our body, then they will start damaging us and breaking down our tissues. And we all know that if somebody has kidney failure, if they don't get